A lot of you that came for the last session remember that I went through a lot of the command line tools. We essentially discussed what bioinformatics is, so that we know it's a really multidisciplinary type of science. It brings together lots of different experts from lots of different from fields. So you have mathematicians, we have computer scientists, we have software engineers, we have evolutionary biologists, lots and lots of people. We're all coming together to analyze this sequence data. So we discussed briefly about how we get the raw sequences from the machines that we use. So there's two different types. We have the short reads, which can be between 150 to 150 base pairs usually. These come from Illumina machines. And then we also discussed very briefly about long read sequencing technology, such as from PacBio. And these can produce kilobase long sequences and help us to reconstruct genomes. I want to say more easily, but there's pros and cons attached to it. So what we do is once we get these raw plain text files, we then proceed to some sort of quality control, and then we process them into what we call BAM files, which is a binary alignment map, which essentially tells us which of the reads are assigned to which part of the genome. Lots of different people across PANGEA are doing different things with these sequences. I, for one, look at resistance. Andy Rambo's group looks at phylogenetics and reconstruction of phylogenies and Bayesian trees and stuff like that. And you know, each of you are working on something different that we use for these sequences. So that's what we looked at last session. As part of this session, Lucy very kindly sent out some emails and asked you guys what you would like us to basically talk about in these sessions. So here are a couple of examples of questions that I was forwarded. So we had a couple of people ask if there's a example data set that they could use for the different software which we talk about in the webinars. Now, whilst we don't have a specific set that we use across all of these different software, there are many available on the internet. When you download Filoscanner, which you can see from Filoscanner GitHub, there's lots of examples inside here. When you download pretty much any software, to be honest, whether that be from the people's GitHubs, or even if you install through the Conda environments or the Homebrew environments like we discussed last session, all of these come with example files which you can use to get to grips with the software. But if you are struggling, then feel free to reach out to any of us and we can maybe point you in the right direction. But certainly if you're looking for a nice data set to use for Filoscanner or for other software, look here and you can find some information inside here. Another question we had were people asking for specific software for us to go through. Now, I'm not an expert in BioEdit or HIV Trace or even Beast, though I have used these software. Perhaps it's not, I'm not the best person to teach these things in a very sort of basic introduction to bioinformatics. So I discussed this with Lucy, and if you want us to go through anything specific after this session, then you can email your thoughts to Lucy, and then we can reach out to other researchers in the PANGEA network and see who's best to deliver these sorts of sessions. And then we can, we're always looking for ideas to help train people and basically make some more webinars. So yeah, you can either post on the forum, there's a link here, which Lucy has also mentioned in the past. Otherwise, you can, just email her with some ideas or anything that you'd like to see, and then we'll try and arrange that for you. So two other questions which were asked, which I am going to go through as part of this basic introduction to bioinformatics. One of the questions that we were asked is, what's the best way to store data files which are generated by PANGEA? So the types of files that we generate include consensus sequences, which are made with Shiver, as well as the BAM files, which is also made with Shiver, actually, I believe. These are the things that we store at Oxford. These are the things that you can request as PANGEA researchers once you have become accredited. We'll go through that in a little bit. And another question is, somebody asked, what is the difference between an alignment and a mapping of raw reads to sequences? So I realized that maybe not everybody fully understands what the BAM file is and how we've got there. So we'll talk a little bit about the differences between an alignment and a specific mapping in a little bit. So let's have a look and see. So there's lots of different data files which we produce and we store as part of whole genome sequence analysis. Really deciding on which format that you want to store is highly dependent on the resources that you have at hand and really what you want to do with it. So I pulled a couple of files that I've made myself using PANGEA data. And you can see here the different types. So we have compressed raw reads, which are FASTQ, which have been gun zipped. I mean, these are very small. They can be just 1 or 2 megabytes. So you can store, hundreds of thousands of these with no real problem, to be honest. When you start getting to unzipping these, they get a bit bigger, and then when you start to make SAM and BAM files, they can span hundreds of megabytes, and that can very quickly sort of start taking up hard drive space. So generally speaking, where I work at UCL, we have access to computer clusters, online computer clusters, or Linux boxes. These generally have between 1 and 2 terabytes of storage, so whatever I'm usually working on, I store on online servers. I mean, outside of the UK, you can rent services like, I'm trying to think of what's a good one to use. It's important to remember that this data, it has to be kept secure, so you really shouldn't be storing any of these sorts of things on Google Drive or anything which can be easily breached, because at the end of the day, we're dealing with patient sequences, and they should be really kept confidential. So if you're looking for a good storage solution, then the minimum that I would probably suggest is keep the FASTQ files. You can keep them compressed, you can keep them uncompressed, keep them on an encrypted hard drive. These don't tend to be so expensive. You can get 500 gigs a terabyte for not so much. That's probably the most cost-efficient way. Hopefully that answered that question. But generally speaking, we try to keep them on an online server. Otherwise, we also have backups on hard drives, which we encrypt with strong passwords. So alignments and mapping. Generally speaking, within PANGEA, I use mapping. So we have a specific reference sequence. So for instance, on the South African task dataset, we know that the majority of these are subtype C. So we have a manually annotated subtype C sequence, which we map all of our raw reads to. But there are instances where you don't have a specific reference genome which you can map to. So in this case, we might use a de novo assembly, which means building larger contigs from raw sequences without a reference backbone, essentially. So I'll try to explain this by using some diagrams. So if you imagine each of your short reads, which comes from the Illumina MiSeq or Illumina HiSeq, which is one of the most commonly used sequencing machines, you imagine each of these fragments are between 150 and 250 base pairs, for example. Using mapping, what we might do is we might have the reference C, the HIV subtype C reference genome, which I mentioned that we use. Essentially, what we do is then we use a computer to essentially slot the reads as if it were a puzzle. So any overlapping sequences will be built into a sort of continuous genome. So say, for instance, you have ACTG, it will find ACTG in the genome and then map to it. When you are assembling without a reference genome, what will happen is it will do this on a base by base I don't know what I'm trying to say. It does it slightly differently. We can look this up a bit later, and I can send you some links to show you the process of how it happens. But essentially, it will build into large contigs, up here I've written, so when you're working with reads with an unknown reference genome, it's assembled into what's called contigs and scaffolds. And then the contigs can then be considered as, you can make an alignment or you can make a mapping to these. So say for instance you build your transcript de novo, so you've arranged each of these as best as the computer thinks. You can then either blast this and see what it's closest to, and then you could use whatever it blasts to most closely as a reference genome, or instead you can actually blast it, see which is the closest, and then you can either do a multiple alignment to get a whole genome, or you can use the same mapping with the new reference that you've found. Now, there's a couple of different issues with the mapping and alignment process. So this is one of the problems that a lot of bioinformaticians have. It's very important that you check the mapping quality scores for your BAM files and SAM files. I mean, you can do this using SAM tools. We went through this looking at the SAM tools view last session. So you can look up last session if you are struggling to do that, or you can have a quick Google. It's not so hard. So basically the mapping score it's an indicator of the sequencing accuracy. So if it has a particularly low mapping score, you might not be 100% confident that that's where the read should go in that genome. It could also indicate something like a contaminant, or just basically an incorrect assembly. There's also issues whereby, say for instance, if you do have a read which is ACTG, it might fit cleanly in this section, but then again it might also fit cleanly in this section, and this is what we call a multi-read. So we have to be aware of things like these as well. And another issue with the alignment process, which is where you can perform a multiple alignment. So if you're not mapping, say for instance you have built your contigs de novo, and then you want to do a multiple alignment to try and form that into a large genome. The alignments, depending on which program you use, they generally can't account for structural changes. So if you have insertions or deletions, it might not necessarily pick those up, which might give you a bit of a dodgy genome. So we do prefer mapping, but if there's no reference sequence available, then you have to see what best fits. But generally speaking, in PANGEA, this is all done for us by the lovely team at Oxford. So we can just request nice BAM files which have been cleaned and everything's been done for you, which is nice. So another question that I had from some people is basically how to use Genius. So Genius is it's a GUI which a lot of PANGEA members use to basically do all of these bioinformatic tools. So it's just a wrapper which you can do everything pointing and clicking instead of having to type in code into a terminal. So it's quite effective for people that don't have any bioinformatics skills or don't want to take the time to learn coding. There are some limitations in that it's kind of expensive. So for academics, like postdocs, professors, etc., it's $450. That gives you a 1-year license. But for PhD students, MSc students, it's only about $200 for a year. And it does have some issues, I'd say, but certainly if you're getting into bioinformatics, it might give you a good taster of which tools work best for the data that you're working with. And as I mentioned in the last series, there's also a 14-day free trial if any of you would like to try it out and basically see how it works. But some of the specific questions that we had revolved around how do you make annotations in Genius. So I've just put together very few slides which go through what Genius looks like, what you can do with Genius, and how to do it. Because when I first bought my license, I was just sort of dropped in the deep end, and I was taught by somebody over Skype, which was a bit difficult. But essentially, this is what Genius looks like. So when you first open up Genius, the latest version is called Genius Prime 2020. They just release a new one every year and slap a different picture on the front of it. This is what it looks like. So you can see over on the left-hand side, this is your file explorer, for want of a better word. So this is all of your working directories and everything that you're doing. You can put it into different folders over on this side. Up the top here, you have what I call quick actions. So these are just shortcuts which go to the most useful things that you might want to do in Genius. So all of your alignments and assemblies done here, all of your trees done here. I'll show you some examples as we go down a little bit more. And then this large window here, this is the working directory. So this is where, this is everything that's inside each of these files. And then each of these little icons, they mean something different. So the little green thing, that's an alignment. This red thing is an assembly, and these are raw sequences, and so on and so forth. So in order to import your reads into Genius, so if you imagine that you have sequenced your sample, and out of the sequencer has popped your 2 FASTQ files. This is what you'll first get when you drag and drop these into Genius. So you can import them as an interleaved file, which is where Genius will match up all of the R1 and R2 files, and it will sort of merge it into one file. So this is for paired end reads. What's important to note here is it will ask you for the insert size. Now, whilst this doesn't make a massive difference if you change this setting, it's better practice to use something like Picard Tools, which will tell you what the insert size is. And the insert size, for anybody that's wondering, is the actual length of DNA or sequence that was sequenced. So this is saying that this was, it was sequenced at 500 base pairs per run or whatever. But anyway, if you want to use something like Picard Tools, then Picard Tools will actually calculate the mean insert size using this function called collect insert size metrics. So in order to install Picard Tools, we went through this during the last tutorial. So you can either use something like Homebrew, you can go to the GitHub, or you can go to the website. Once you've installed it, you essentially, you can use what's written here. So this is just a small set of instructions ready to get this. So once you've downloaded or installed Picard Tools, you get a jar file, which is called picard.jar. So java-jar tells the computer essentially you want to use Java to open the jar file. You point it towards the jar file, which is picard.jar, and then you give it the test that you want it to run, which in this case is collect insert size metrics. And then you give it your BAM file, which give it BAM file, and then you ask it to output size metrics and the histograms, and it will give you something which looks like this. So you can see really if we use something that was, the default is 500, but really we don't have many insert sizes of 500 base pairs. So we'd actually probably use something closer to over here, which is like 225. So once you've imported your raw reads as an interleaved file, it will look like this. You can see what Genius has done. It has paired these up. It's moved all my annotations, which is kind of annoying, but those top 2, you can see they're the same length. So you might have a forward and reverse read here. In this second part, you have a forward and reverse read, same. If you wanted to leave it as 2 individual files, then you could still do assemblies, but it takes a little longer because you have to do them all individually. There's no workflow to do that in Genius. So we talked about the differences between alignments and mapping. This is just an example of how to do those assemblies in Genius. So you can see up here in this quick actions bar sort of thing, we have the alignments and assembly. When you click on that, you have, you can do your multiple alignments, you can map to reference, do de novo assemblies, align whole genomes, so on and so forth. The ones that I tend to use most often is map to reference. So we're using that subtype C reference, or you can do multiple alignments. I do this before building my trees. So once you're clicking on map to reference, you get something which looks like this. It looks a bit scary at first, but it's not so hard to get your head around. Essentially, you tell it what your reference sequence is. So this AF411967, this is one that we, or AN-DARASH rather, manually annotated. So it includes all the different drug resistant mutations and stuff like that. You can then ignore this part. It's normally blanked out, but you can, it's fairly straightforward. You can change it as however you want. This is to do with the names. This one here is grayed out. This is if you want to do lots and lots of mapping to references. You can highlight a load of files, and then if you select this, it will actually map each file separately to the reference that you've selected. Or you can click on this drop-down bar, and in Genius it will give you an option to choose what it thinks is the best reference. So, say for instance you have a mixture of subtype Bs and subtype Cs, you can it will decide somehow if it wants to map it to the subtype B reference or the subtype C reference. There's lots of different mappers. So if you click on here, it opens up this this drop-down menu. So the Genius mapper is a proprietary mapper. I always find it takes absolutely forever to run, even like a high-end Mac. So I have quite a high-end Mac. It just takes forever. I don't know why. Ones which I would recommend include Bowtie 2, which is very fast, or BBMap. These are both very good mapping tools for short reads. I think we used BWA last time, so in the first tutorial we used BWA. That's my preferred mapper, but if you want to use Genius to do your mapping, then I would recommend maybe Bowtie or BBMap. So once you've selected either of these, we'll come back to this screen. Depending on the specifications of your laptop, you can change some of these settings so it uses more or less memory. So sensitivity is quite important to toggle. I think the default is medium sensitivity, which Genius estimates is a fast running. It uses less memory, so it's meant to be faster. I've not found massive differences between medium sensitivity, medium to low, high sensitivity. I mean, it depends if you're getting dodgy looking results. If you're getting dodgy looking results using these mappers, you can use a higher sensitivity and see if that changes anything. So once you've selected your mapper and your sensitivity, usually you can leave most of this to default settings. But if you do want to change anything, then here are the results. I mean, it's the behavior of what you want to do with it. So you can change the assembly name so that it's easy to find. If you're doing downstream analysis, once you've exported this, you don't really want your BAM file or whatever to say this assembled to this. So you can just change this to the name of the reference sequence or the name of the original sequence. It's up to you. Assembly reports can be useful if you're getting dodgy looking assemblies with low coverage and you're expecting higher coverage. If you save this, it goes into like the default directory, which on a Mac is in your home directory. I'm not sure on anything else, but when you first open Genius, it asks you where it wants to save everything. So you can also save a list of unused and used reads. So when you're making an alignment, your BAM file tends to be quite large. You remember that we went through a deduplication process, and then we removed any unmapped reads during my last tutorial. So say, for instance, you have, I don't know, human sequences still in the BAM file. You can remove these by just saving a list of unused reads. If you need to do any trimming, you can toggle this here. So for the subtype C task samples that we made BAM files for, we used to trim off a certain number of bases which corresponded with the PCR method that they used. I don't remember what these were because we use the PANGEA BAM files now. But yeah, you can do trimming here, and then here you can output the minimum mapping quality. So say for instance, if you need a piece of software which will only accept a minimum mapping quality of, say for instance, 30, you can force the BAM file to read 30 each and every time, which is a bit strange and a bit misleading, but okay. So once you've actually made your assembly, so what we've done is, if we go back up again, we've imported our reads, which are now here. This is an interleaved file, so it's already added the forward and reverse reads to one file. You can select that by clicking once, and then you click on Align and Assemble, and then click on Map to Reference. We've gone through this, we've left most of the things as default, except we've changed the mapper to bowtie, and we've changed the sensitivity to medium. That's what I usually do. You can also change how many iterations you want to run as well. It defaults to 5, but if you have not so much memory, or if it's going to take a long time, you can change this to like 2 or 3 or whatever you want. Once you've done that, this is what the BAM file looks like. So you might remember last time we used something called IGV, which is the Integrative Genome Viewer, to look at the BAM file. Genius actually has quite a nice BAM viewer built in. So once you have made your BAM file, you'll remember I showed you the files. They look like this with the 3 red lines, I guess. Once you click on this, it will open the alignment, which looks like this. So you can see this is a map of coverage. This is the annotated reference sequence that we've used. These are all the manual annotations and the drug-resistant mutations that Andreas has put in for us here. And then this is where it's mapped all the reads. And the mapping of the reads corresponds to the coverage. So where you can see very little or no coverage, you can also see that there were no reads there to map to. So one of the specific questions I had about Genius was, how do you add annotations? So say, for instance, you have made a de novo assembly, you blasted it, you found that it matches to, I don't know, a subtype B strain of HIV, and then you download it, and then it has nothing on it at all. So let's give an example of how to add an annotation. It's quite simple to do. So here's an actual example of work that I've been doing recently. So we're interested in one of these integrase mutations at position 74. So we look for the integrase gene, and say, for instance, it will be, I don't know, position 74, let's call it here. I don't remember exactly where it is. So what we do is we can add a custom annotation to our reference sequence by clicking on the reference sequence in the window viewer. You then find the location that you want to add the annotation to. So the easiest way of doing that, I guess, is just to click on the integrase gene. What that will then do, it will highlight between here and here. So say, for instance, it might say the integrase starts at, I don't know, position 4,230. If I know that the position is 74, and I know that each of these is one codon, I can just multiply that, with a calculator. So I can say, for instance, say that position 74 is the start of integrase time plus 3 times 74. And that takes me here to position 4,433. So all I do is drag across and select this CTG, which is my position 74. And then you click on Add Annotation, which is just above, I cropped it off. It's just above here, basically. It looks like this. So select Annotation, then it will open up this. You can then just give it its name. You can change it to a type. So each of these are different things on Genius. So each of these purple ones are called motifs. Each of these are proteins. Each of these are genes. So you can change it to whatever type you think it might be. And then you can also say if it's a forward or if it's a reverse direction mutation or motif or whatever you want. So in my case, it's a forward direction mutation. So I typed in L74I, I changed this to motif, and then I click OK, and then you can see I've added it here. So that will forevermore be in here. So if I want to use Genius to look for drug-resistant mutations, I can make my assembly, and then I can quickly look down and I can see if any of the reads have mapped to this specific resistance mutation. And you can add and take away anything you want. You can do multiple alignments of lots of different genomes in Genius. Add annotations across. You can transfer annotations between 2 different genomes. It's very intuitive in how it works. This is an example of what I mentioned about the de novo mapping. So you can also do de novo mapping in Genius. What you want to do is import your two raw reads, as we discussed before. And then this time, instead of clicking map to reference, you click on where is it? You'll click on align and assemble, and then here we go. You'll click de novo assembly, which is this one here. What that will then do is build lots and lots and lots of contigs or scaffolds, and then you can blast those to see what they most closely map to, or you can try and do a multiple alignment, but that doesn't always give the best results. So I think the best thing to do is find one of your longer contigs, blast it. You can do that inside Genius as well by looking like this. So here's an example of one that I did a de novo assembly for. So you can see that we have so many different contigs. You can just right click on these and click on blast. When you click on blast, it will ask you for various options like how many matches do you want, the amount of time that you want to spend searching and things like that. And then what it will do is it will give you this window here. So it shows that some of my contigs, the closest match is, say for instance, this KM049902. So what you can do is you can actually download that within Genius to your Genius. You can literally just right click on this, click copy document, and then you can copy it to, say for instance, the same folder that this is. You can then map some of your contigs to that closest reference in order to get a whole genome. Or you can just ask Oxford and they'll give you the BAM files which they've made using Shiver. So I mean, here's an example of the same files. On the left is the de novo assembly that I did, and on the right is a reference-based mapping that I did. So I mean, they kind of look similar, despite the fact that they've not used the same reference genome. So this is the one that we've used our AF411967, which is our manually annotated subtype C, and this is the KM049902, which is the blast result. So you can see down here that the reads, they do look slightly different because these have all been built by essentially guesswork, I guess, and these have been mapped specifically to a genome, but they're not massively different. I guess they'll probably give you the same answers, but this is just an example of what you would do if you didn't have something to map specifically to, I guess. You can also do tree building within Genius. So in the last session, we went through my maximizer, maximum likelihood method of building trees. Within Genius, there's various different tree builders. They have, again, their proprietary tree builder, a consensus tree builder. They have Mr. Bai's, Gali, Fast Tree. They have an older version of Raxamil. I don't know what this one is, and FireMill, which is quite fast. So let's go across to Genius, because I have Genius open here. So maybe we can do a bit of a hands-on thing. So Genius can handle loads and loads and loads and loads of sequences at one time. So here you can see that I've imported something like 10,472 PANGEA sequences into Genius. I mean, using Genius is largely dependent on your laptop, because when we're doing all of this command line code, we're often exporting this to a server somewhere, which is much faster, like infinitely faster. This is trying to load 10,400 sequences, but it's also running some other stuff in the background, which is why it's looking a bit slow and hanging at the minute. But largely dependent on your laptop. Let's let that run. I'll keep talking about tree build at the same time. So anyway, I was saying once you have made an alignment, you can then click on that, and then you can use the tree building icon. The tree building icon then opens up this window, You can then select, all the different settings that you want. So say, for instance, if you wanted to use RaxML from within Genius instead of typing out code each time, you can literally click on RaxML and you can change all of the models. You can change this from gamma to, yeah, that's crashed. Okay, let's just leave that. You can have a look and play around. It has lots of different mods. So it has GTR, GTR plus R, GTR plus R plus I, etc., etc. And my laptop's really not happy now. Sorry. Different algorithms. So within RaxML, generally speaking, we want to be doing a certain number of bootstraps. So I said during my last session that if you want a very fast tree, you can do a lower number of bootstraps. For publications, we want to be doing sort of between 500 and 1,000. You can change all of those sorts of settings from within here. You can also give it additional options. So say, for instance, when you're typing your code into the command line, you can also add anything that you want to in here as well. So say, for instance, you have gone into Genius, you've imported all of your files, you have made alignments, you have made some trees. If you want to export those files to use anything that's not supported by Genius, it's quite easy to do. You can just highlight all of the files that you need to, and then basically click on File, Export. If you want to export single documents, then you click on Selected Documents. For some reason, even if you have selected lots of files, this only ever seemed to export one document for me. So I always click on Batch Export. So say, for instance, with the PANGEA files, the ones that I was working on that I just tried to show you, which are still not working. I exported all of those to our server, and then I can run, the resistance pipeline on them separately. So what I did was I selected all of them, I hit File, I hit Export, I hit Batch Export. It will then basically ask you where you want to export them to, what file format you want to export to, and then the location. So say, for instance, I wanted to export a BAM file, but I only wanted to export the aligned sequences. You can export a BAM file to a FASTA file, and it will do the conversion for you from within Genius, which is quite nice, which means you don't have to go typing code into the command lines. So that's one thing Genius really excels at, is converting between lots of different formats. But anyway, once you've exported all of these, you can do downstream analysis with command line tools or your own custom scripts or whatever you want. Okay. So I mentioned right back at the beginning of the webinar that we are obviously trying to put these sorts of webinars together to help teach people that want to learn. If you want to learn something specific in upcoming sessions, then please feel free to let us know by contacting Lucy, and then I guess she will do her very best to figure out who's the best person to deliver that, and then we can try and teach as we go. And then that means that everybody feels included, everybody's up to speed, and everybody can do really good science and help PANGEA be a big wonder, I guess.